Hi, my name is Anderson Vargas, and I'm going to be talking about mitosis. Mitosis is a part of the M phase, and the M phase is a small part inside of the cell cycle. The first thing that happens inside of the cell cycle is interphase. The first phase inside of interphase is the G1 phase. During this phase, it's a growth phase, so the cell is maturing. And that's basically all that happens inside of the G1 phase. But there's a really big thing called the G1 checkpoint. The G1 checkpoint checks for damaged DNA. It's running through all the DNA and it's making sure everything's good and where it's supposed to be. But the big thing that we have to know about a checkpoint is a checkpoint happens before an irreversible process. So, moving on from the G1 phase, we have the S phase. The S phase is the DNA replication phase. So what's happening is all the genetic material inside of DNA, the cell, is basically doubling. So that's all that really happens inside of the S phase, nothing too fancy. And then it moves into the G2 phase. The G2 phase is another growth phase. Within this phase, it has another checkpoint, as in the as the same as G1 checkpoint. But what's different about the G2 checkpoint is it also checks for damaged DNA, but it also checks for incompletely replicated DNA that took place in the S phase. So again, this is checking to make sure everything's good, but what this one is checking for is damaged DNA and for anything that went wrong inside of the duplication phase and anything that's wonky. If something is wonky, it will be sent off to be repaired or to the lysosomes to basically be eaten. But if everything checks out, then it will move on to the M phase. The M phase is where we finally move on to mitosis and cytokinesis. Now, this is a zoomed in version of mitosis, of the M phase, which has mitosis and cytokinesis inside of it. The first thing that happens within the M phase is mitosis, which is the first thing that happens inside of mitosis is prophase. During prophase, we have our chromosomes, and we have our nuclear envelope, and we have our centrioles. What happens is the centrioles begin to go around the cell, and they go into opposite ends. So we're going to have one centriole up here and one centriole down here. These are our poles. So now that they're moving towards each other's poles, the nuclear envelope is beginning to be eaten or break down. This is the nuclear envelope. One way to describe the nuclear envelope is a plastic baggie. I'll get more into that later, but just kind of keep that into mind. So, while the nuclear envelope is breaking down, the chromosomes are condensing and the spindle fibers start to form on the very ends of the centrioles. So now, moving on from prophase is metaphase. Metaphase is a really big phase because all the chromosomes align. Basically, I only have 2 chromosomes to simplify what's all happening, but we're going to have a lot more chromosomes that are on this little line. And to be exact, 46 of them are in this perfectly straight line that are dead center of the centrioles. So, also the spindle fibers fully develop and they begin to start fly fishing, or they begin to shoot out their little nets or whatever you want to call them, or strings in a way, to catch the centromere of the chromosomes. Up here we see our centromere, that's this little thing, and they're basically pulling up, they're fishing, grabbing onto this, and they're tying kind of a shoelace knot to them. So those are locked on, those are good and tight, but before we can move into the anaphase, we have something called a sac checkpoint. What the sac checkpoint is it's the spindle assembly checkpoint. Basically, it's just making sure all of those shoelaces are nice and tight and where they're supposed to be, because if we have any loose shoelaces, that's where we can get some messy pulls, which are about to happen in anaphase. During anaphase, we have the sister chromatids, which are separating at the centromere. So what's happening is they're taking the centromere and they're splitting it in half. So here we can have our chromatids, which are being separated. So now they're separated, and this is really the biggest buildup from everything happening within G1, G2, and the S phase. It's all moved into this part where the beginning of the separation of the cell is happening. So once everything's done in anaphase and everything checks out, we'll move on to telophase. So telophase is where the nuclear envelope begins to reform. That's the little teeny line that we have. And the chromosomes begin to decondense, whereas in prophase they were condensing, so now they're decondensing. They're basically going to their default selves, and we begin to get something called the cleavage furrow. The cleavage furrow is just something that's totally pinching at the cell itself to split it in half in a way. So going back to the plastic baggie analogy that I was making with the nuclear envelope, is that why do we need to remake the nuclear envelope? It's because we're trying to keep all of the genetic material together. So if you take a plastic baggie and you fill it with water, you have just a plastic baggie with water. You can tip the plastic baggie any way and the water won't fall out. Now if you take that same plastic baggie with water and you put it inside of a bucket which is filled with water, you'll have just a bucket of water with a plastic baggie with water. What's happening is that water inside of that plastic baggie isn't flowing outside of the plastic baggie and into the bucket of water. So basically, this is just keeping all the genetic material safe and sound inside of itself. Now, what's on the outer rings is a constant flowing form of cytoplasm, which is just moving everywhere. So if we did not have our nuclear envelope, that would kind of make everything fly everywhere. So now that telophase is done and that we've pinched the cell in half, we move on to cytokinesis. Cytokinesis is the last step inside of the M phase. This is where our cells have completely separated, and now we have a total of 23 chromosomes in both of these cells, and these are the daughter cells of the parent cell. So these are the daughter cells in the prophase, or this is the parent cell, which began in prophase. So the biggest thing to see about the cytokinesis is the cytoplasm separates. This is the cytoplasm. It's that constant flowing liquid that's inside. This is the cytoplasm. So the cytoplasm has separated and all the genetic material inside has separated. So that's basically all we need to know about mitosis. Thank you for watching this video, and my name is Anderson Vargas.